Good morning students. In today's class, we will see about 2 mark and 3 mark questions in Principles and Process of Biotechnology in Plus 2 Biobotany. First question is, define cymology. It's in page number 69, your Biobotany book, page number 69. Under the topic Method of Biotechnology 4.2, come to the 10th line. The study of fermentation, its practical uses is called cymology. The study of fermentation and its practical uses is called cymology. Second question, define bioreactor. Page number 69. Bioreactor is a vessel or a container that is designed in such a way that it can provide an optimal environment in which microorganisms or their enzymes interact with a substrate to produce required product. Second point, in the bioreactor, aeration, agitation, temperature and pH are controlled. Third question, name the downstream process in fermentation. Page number 69 and 70. Under the heading downstream process, open the bracket. The downstream process in fermentation are underline distillation, centrifuging, filtration and solvent extraction. The next question, define single cell protein. It's given in page number 70. Under the topic 4.2.2 single cell protein. Single cell proteins are dried cells of microorganisms that are used as protein supplement in human foods and animal feed. Point number 1. Single cell protein offers an unconventional but plausible solution to protein deficiency faced by the entire humanity. Close the bracket. Next question. Name the microorganism used in single cell protein. Page number 71. Answer is in page number 71. The microorganism used in single cell protein are bacteria, Methylophilus, Methylotropous, Cellulomonas, alkaligenes. Fungi, Agaricus campestris, Saccharomyces cerevisiae, Candida utilis. Algae, Spirulina, Chlorella, Chlamydomonas. Students, each one, that is bacteria, separate 2 mark question. Fungi, what are the fungal organism used as SCP and what are the algal organism used as SCP? Each one organism, that is in bacteria, fungi and algae, are separate 2 mark question, and together you study for 2 mark or 3 mark question. The next question is 6th question. What are the primary metabolites? Name the types with example. Page number 70. Students, take page number 70. Primary metabolites. In page number 70, microbial metabolites. It is given under the heading microbial metabolites. Microbes produce compounds that are very useful to man and animal. These compounds are called metabolites, and it is divided into 2 groups, primary metabolite and secondary metabolite. Metabolites produced for the maintenance of life process of microbes are known as primary metabolites. Example, ethanol, citric acid. Secondary metabolites are those which are not required for the vital life process of microbe but have value added nature. Example are amphotericin, penicillin, streptomycin, ariomycin, tetracycline. Any 2 example you can write. Next question. List out the application of SCP. Page number 71. It is given under the heading application of single cell protein. It is used as protein supplement, cosmetic products for healthy hair and skin, excellent source of protein for feeding cattle, birds and fishes, and in food industry as aroma carriers, vitamin carrier, emulsifying agent. It is used in industries like paper processing, leather processing and foam stabilizers. Next question. Define recombinant DNA technology. It is given in page number 71. Page number 71. Under the heading Genetic Engineering 4.3.1, genetic engineering or recombinant DNA technology or gene cloning is collective term that include different experimental protocol resulting in the modification and transfer of DNA from one organism to another. Close the bracket. Next question. Define insert. Page number 71. Under the topic 4.3.2, steps involved in recombinant DNA technology. First point. Isolation of a DNA fragment containing a gene of interest that need to be cloned is called insert. Close the bracket. Define PCR. PCR means it is given in page number 71. Answer is in page number 71. PCR means polymerase chain reaction. PCR, polymerase chain reaction, is a common laboratory technique used to make copies of particular region of DNA. 11th question. Name the genetic engineering tools. Page number 72. It is given in page number 72. Genetic engineering tools. Under the heading Tools for Genetic Engineering 4.4, in that paragraph, the most important enzymes, the basic tools, the first line, that is in the page number 72, right side first line. The basic tool are enzymes, vectors and host organism. The most important enzymes for genetic engineering are the restriction enzyme, DNA ligase and alkaline phosphatase. Close the bracket. The next question. Define Rdna. It is given in page number 71 and 72. Rdna or recombinant DNA. Last line of page number 71. Rdna means it is formed by insertion of DNA fragment into the carrier molecule called vector that can self-replicate within the host. That is, gene of interest plus vector DNA is called Rdna. Gene of interest combined with the vector DNA is called Rdna, recombinant DNA. Next question is, what is restriction site? It is given in page number 72, restriction site. Under the heading Restriction Enzyme 4.4.1, in that, after 10th line, a restriction enzyme or restriction endonuclease is an enzyme that cleaves DNA fragment at or near specific recognition site within the molecule known as restriction site. That is the cutting point of the restriction endonuclease in a DNA. Cleavage site is called restriction site. Close the bracket. Next is define restriction enzyme and its type. Page number 72. It is given in page number 72. Restriction enzymes. Page number 72, under the heading Restriction Enzyme 4.4.1. The two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated in 1963, and the two group of enzymes are, after that paragraph, first A, point A, exonuclease are enzyme which remove nucleotide one at a time from the end of a DNA molecule. Then second one B, endonuclease, exonuclease and endonuclease. Endonuclease are enzyme which break the internal phosphodiester bond within the DNA molecule. Close the bracket. Then next question, 15th question. Define palindromic repeats. It is given in page number 73. Palindromic repeats. Palindromic repeats means, that is page number 73, under the heading Restriction Endonuclease Molecular Scissor, in that topic, third paragraph from the bottom, third line, palindromic sequence or palindromic series means that the sequence in both DNA strand at this site read same in 5' to 3' direction and in the 3' to 5' direction. That is given in the blue color box. A symmetrical repeated sequence in DNA strand from 5' CATTATATATG, that is guanosine, guanine, adenine, thymine, thymine, adenine, thymine, adenine, thymine, 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 guanine, 3'. Likewise 3' to 5' direction. Close the bracket. Then next question. Name the three main classes of restriction endonuclease. Answer is given in page number 73. Three main classes of restriction endonuclease. The three main classes of restriction endonuclease are in the second paragraph, page number 73, second paragraph, left side, second paragraph. There are three main classes of restriction endonuclease: type 1, type 2, type 3. Type 2 enzyme is preferred for use in recombinant DNA technology. They recognize and cut DNA within a specific sequence. First one. Then second, in the third paragraph, the restriction enzyme hint 2 always cut a DNA molecule at a point of recognition of specific sequence of 6 base pair. Then in the right side, E. coli. E. coli is from Escherichia coli, strain RY13, and first endonuclease 1 to be discovered. Close the bracket. Next question, 17th question. E. coli define. Page number 73. In page number 73, right side it is given E. coli. E. coli means Escherichia coli restriction endonuclease enzyme 1. E. coli is from Escherichia coli strain RY13 and the first endonuclease 1 to be discovered. Close the bracket. E. coli. That is the definition or full form of E. coli. Then 18th question. Blend end and cohesive end differentiate. Page number 73. Blend end, cohesive end differentiate. First blend end. Page number 73, right side, second paragraph. The exact kind of, open the bracket, students, open the bracket. The exact kind of cleavage produced by a restriction enzyme is important in the design of a gene cloning experiment. Some cleave both strands of DNA through the center, resulting in blend or flush end. Close it. This is known as symmetrical cut. And there are some next line is for cohesive end. Some enzymes cut in. There are some enzymes which cut in a way producing protruding and recessed ends known as sticky or cohesive end. Such cuts are called staggered or asymmetrical cuts. The next question, 19th question. Define DNA ligase. Which organism it is isolated? Students, answer is given in page number 73. DNA ligase, under the heading DNA ligase, open the bracket. DNA ligase enzyme joins sugar and phosphate molecule of double-stranded DNA with a 5' phosphate PO4 and 3' OH hydroxyl group in an adenosine triphosphate dependent reaction. This is isolated from T4 page and it is one more question, very, very important. The next question, define alkaline phosphatase. Which organism it is isolated? Question number 20. Alkaline phosphatase. It is given in page number 73 under the heading 4.4.3 alkaline phosphatase. DNA modifying enzyme. Alkaline phosphatase is a DNA modifying enzyme Adds or removes specific phosphate group at 5' terminus of double-stranded DNA or single-stranded DNA. Thus, it prevents self-ligation. And this is isolated from bacteria and calf intestine. The next question is, this calf intestine and bacteria is isolated. This isolated from bacteria and calf intestine. This is one more question. This answer is very, very important. The next question, define vector, page number 74. Under the heading 4.4.4 vector, it is given answer is from the second line end. A vector is a small DNA molecule capable of self-replication, and it is used as a carrier and transporter of DNA fragment which is inserted for cloning experiment. Vector is also called cloning vehicle or cloning DNA. Close the bracket. The next question, 22nd question. Differentiate two types of vector. Differentiate two types of vector. Vector are of two types, cloning vector and expression vector. Answer is given in page number 74 under the heading vector 4.4.4. In that, cloning vector, first answer is cloning vector. Cloning vector is used for the cloning of DNA insert inside the suitable host. Then expression vector. Expression vector is used to express DNA insert for producing specific protein inside the host. Next question. Define plasmid. 23rd question. Define plasmid. Page number 75. In page number 75, types of vector is given below that heading plasmid is given. Plasmid are, open the bracket students, plasmid are extra chromosomal self-replicating circular DNA molecules, double-stranded, that is DS. DS means double-stranded. Double-stranded circular DNA molecules found in the bacterial cell in addition to the bacterial chromosome. Plasmid contain genetic information for their own replication. Close the bracket. This is define plasmid. Then 24th question, define PBr 322. Page number, answer is given in page number 75. In that, students, PBr 322 means third line, from third line. P denotes plasmid. Br respectively the names of scientists Bolivar and Rodriguez who discovered the plasmid, and the number 322 is the number of plasmid developed from their laboratory. Close the bracket. The next, define Ti plasmid. 25th question, define Ti plasmid. Answer is given in page number 75. Ti plasmid. Ti plasmid is found in Agrobacterium tumefaciens. Point number 1. The plasmid carries transfer gene which help to transfer tDNA from one bacterium to other bacteria or plant cell. Close the bracket. The next question, 26th question. Name the two methods of gene transfer in plants. Answer is given in page number 76. In page number 76, the two methods of gene transfer in plants are, in page number 76, last two lines, direct or vectorless gene transfer 1, indirect or vector mediated gene transfer 2. Next question is, name the different types of vectorless gene transfer method. Page number 76 and page number 77. Students, in page number 76, there are 3 methods, and page number 77, 2 methods. Page number 76, 3 methods are chemical mediated gene transfer, microinjection, electroporation, liposome mediated gene transfer, and Biolistic method. Totally 5 methods. Gene transfer method. Vectorless gene transfer method. Totally there are 5 methods. Chemical mediated gene transfer, microinjection, electroporation, liposome method and Biolistic method. Next, 28th question. Name the chemical used in vectorless gene transfer. Page number 76. In page number 76, under the heading Direct or Vectorless Gene Transfer, Chemical Mediated Gene Transfer, one heading is given there, no? In that, polyethylene glycol, PEG, and dextran sulfate, these are the two chemicals, polyethylene glycol, PEG, dextran sulfate are the two chemicals used in vectorless gene transfer, right? Next question, define electroporation. It is given in page number 76, students. Page number 76, electroporation. Electroporation means a pulse of high voltage is applied to protoplast or cell or tissues which makes pores in the plasma membrane through which uptake of foreign DNA occurs. Close the bracket. I hope you all marked. Okay. The next is lipofaction. Define lipofaction. Question number 30. Define lipofaction. Page number 77. In page number 77, liposome mediated, under the heading liposome mediated method of gene transfer is there, no? Liposomes are phospholipid. Liposomes means that are phospholipid. Phospholipid vesicles are used in gene transfer. First point, phospholipid vesicles are used in gene transfer. Second point, the gene or DNA is transferred from liposome to vacuole of plant cell. Third point, last line, come to the last line, liposome and tonoplast. Tonoplast means the outer covering of vacuole is called tonoplast. Liposome and tonoplast of vacuole fusion resulted in gene transfer. This process is called lipofaction. Close the bracket. The next is define biolistic method. Question number 31. Define biolistic method. It is given in page number 77. In page number 77, biolistic method. Under the heading Biolistic method. Foreign DNA is coated on the surface of coated with minute gold and tungsten particle and bombarded into the target tissue or cell using a particle gun. Then the bombarded cells or tissues are cultured on selected medium to generate plants from transformed cells. Right? Students, so far we discussed about 31 questions in in principles and process of biotechnology. I hope you all marked these 2 mark and 3 mark questions in your biobotany 4th lesson. Students, please study these questions and write 2-3 times. Well prepared for the examination and get ready for the examination. All the best. God bless you. Thank you.